Now we're going to zoom into one particular type of the passive transport, which is going to be the movement of water. And movement of water is also defined as osmosis. These are key concepts that you can apply to your study in chemistry, physics. Osmosis is an application of how water tends to move towards an area that has a higher amount or abundance of solids. So this means that the question I posed to you all on Tuesday is the water will tend to now want to leak out of the cell and thereby causing the cell to shrivel down, right? So that scenario that we answered in that discussion question is literally right here, the one on the bottom, right? This is the one where we call it hypertonic, and the term we use to describe a condition where the environment has more stuff than the internal compartment of the cell, we call that hypertonic. You flip that around, and if you have more solids internal to the environment and internal to the cell, we call that hypotonic. And as a result of that, water tends to swell into the cell. Right? So that can lead to a condition known as lysis. Okay? And lysis or lysis, however you like to say it, is when you rupture and burst open a cell. This is the same phenomenon that most cells use to kill off other cells. So when you're thinking about a virus infecting a cell that's healthy, and now that healthy cell becomes bad, how do you kill that bad cell? Well, one mechanism of killing off these bad cells is by utilizing water and then the bursting effect of lysis. The first type on this chart here is isotonic. And the word iso means equivalent and balanced out. And therefore, in an isotonic solution, the net water entry and the net water exit is the same. So, in other words, think about which one of these conditions your cells would prefer. Which one of these conditions do you think is the healthiest for your cells to be? Right? My bet is probably isotonic. But do you think that we can maintain a perfect tonicity every day, every month, every week? All right, so now active transport, when we take a look at this, is where you utilize energy. And there's a lot of examples in the different organisms, but in our bodies, in the human body, one of the most notable classic iconic example of an active transport that utilizes energy is also known as a sodium-potassium pump. And you're probably thinking, Lynn, what the heck is this? Well, sodium-potassium pump is an ion pump, right? That means it moves sodium. Sodium is Na. And then potassium is the famous K plus, right? Like the reason why we all eat bananas. And sodium and potassium are both two cations, right? You guys see that? It's both positively charged ions. So you're thinking, well, if it's a positive and negative, they attract. But what about a positive and a positive, right? They should repel. So an active transport is when you move something against the gradient. Remember, we defined all this on Tuesday already. So if you don't remember this, you need to rewatch the video. And when you go from an area that is low to an area that's high, what you are doing is you are climbing up the hill. Right? Remember the analogy I gave you guys where you have a slide, right? You go to a playground and then you go up to the top of the slide, and then you have this long slide where you can slide right down, right? And then you're happy. Well, in active transport, what you are doing is you're going up the slide. In passive transport, you fortunately get to go down the slide. Okay? So that's a way to think of this. On the right-hand side, you can see this picture, and I'll illustrate how this sodium-potassium pump works. What's the significance of the NAK pump? Well, the NAK pump is found in all nerve cells in the human body. Right? So if you have a nerve cell, which you probably have, that are enabling you to process information in your brain, control your arm and leg, maintain sensation of temperature and itching and pain, those cells rely on this pump to generate a charge along the membrane. And if you look at this picture, it might be a little confusing, but it's not that bad, all right? So let me go ahead and zoom into the story just a little bit for you, all right? So you can see right here on step number 1, the NAK pump begins by opening up to the internal side of the cell and allowing 3, right? 3, not 1, not 2, but 3 sodium ions to bind into its chambers, right? You guys see that there's these 3 square-like indentations that allow the sodiums to bind onto. So it opens to one side first. That will then trigger the utilization of energy in the form of ATP. So step 2, ATP comes along, and now you take advantage of that phosphate, that's very, very unstable phosphate, to charge up this protein and cause a change in shape. Right? I know you can't really see this, but this little orange protein that you see, this is like the door. And when the ATP affects it, it's going to change the shape. Now you can see clearly 3 sodiums have bound onto that chamber. It's going to then open up to the outside and now allowing the sodiums to move out. Right? But remember, it's going against the gradient. That means normally there's more sodiums on the outside. All right? So that tells you which side has more stuff. And then the 4th step is now that since it's opened up on the outside, these little round pink potassium ions can bind onto the chambers where there's circular indentations and now close down, open up internally again, and release those potassium ions. So let's summarize what I just said. Number 1, this is an active transport that utilizes ATP. Number 2, this pump moves sodium out to the cell and potassium into the cell. Is it an even ratio? No, right? So that's why if you think about biology at a molecular level and even at a cellular ionic level, not everything is a fair trade, right? We all hear the saying, and it's overly used term, life isn't fair, right? We all have different starting points, and of course, per all of our environment and our prep and your ability to start off where you begin is going to be very different, right? So your starting line is going to be very different, and so even at an ionic level, sodium, potassium, they're not equivalent. It's every 2 potassiums, you get 3 sodiums. Okay, so it's not a fair trade. All right, so let's keep the ball rolling. So now, to make this picture complete, the process of energy creation is also known as cellular respiration. Cellular respiration is the fancy way for saying metabolism, right? And we've already established the fact that we all need energy to survive, and energy is utilized in all facets of biology, whether it be contraction of muscle, moving of ions, transporting of signals and hormones. But this energy is oftentimes stored in these bonds between phosphates. All right, so let me just highlight that key phrase here. So ATP, right, ATP has 3 phosphates because it's triphosphate. And these phosphates, right, these inorganic phosphates are what contain the energy. Okay, so it has 3 of them. It has 3 pyrophosphate, or this inorganic phosphate. Okay, these 3 P's. So what you're looking at on this picture here, this chart, is the metabolic pathway in all of biology. Okay, this is the summary, and it's a lot more complex than it looks, but this is literally a snapshot of everything. So you have one reaction known as glycolysis. This is what we call anaerobic or non-oxygen requiring metabolism. And you make a little bit of energy, which is great, but you don't make a whole lot. And the starting product, the starting reactant of this whole reaction is glucose, is the famous carbs that we all love to eat. The end product is a 3-carbon molecule called pyruvic acid. All right, so all you need to know for this class, right, this is not chemistry, is you need to know at least you've heard of this terminology before, right? So when somebody says glycolysis, when you go to the gym and you're working out and you're lifting weights and you're trying to generate a quick 1 rep max or your PR, you are utilizing glycolysis to burst that energy. When you're going for a long form exercise and you have the ability to respire and drink water and stay hydrated, you are then tapping into some of the other reactions. All right, so glycolysis is glucose to pyruvate, and then after that, this energy in the form of NADH, ATP, mainly ATP, gets shuffled into the mitochondria, and this big globular structure right here is the mitochondria, and now it takes over with another couple of reactions. The Krebs reaction, which is basically an opportunity to give off some CO2. The CO2 gets given off right here. And then we have the Krebs cycle, also known as citric acid cycle, which is about 10 steps. And this utilizes different intermediates, mostly amino acids, to generate more energy, building products like NADH. And we'll talk a little bit about this guy right here. NADH is a cousin to ATP. They all are nucleotides, and these have this ability to generate and break out as energy. Okay, so it's a carrier, if you will. Think of an energy carrier. And then the final reaction is called the electron transfer chain, and this is specific to a set of proteins and pumps that will then make most of the ATP. All right, so your goal here is how do you get from sugar, right, the carbs that you eat, to ATP? Well, this is the factory. It goes from glycolysis, 1, Krebs cycle, 2, CAC, Calvin cycle, Krebs cycle, 3, and number 4, electron transfer chain to make the most ATP. And if you're curious, right, there's a natural numeric value. So for every 1 glucose that you take in, you can make an upwards of 30 to 32 ATP, which is pretty awesome, right? So if you think about it, you can actually get a value. 1 glucose yields about 30 to 32 ATP. All right, so it all comes down to breaking bonds, taking advantage of ATP, and then stripping that off to make ADP. And there are these other high electron carriers, which I pointed out, like NADH, and you'll get a chance to hear a little bit more about these guys in your reading in your Learn Smart. So here is going to be where things get really fun. We're going to look into glycolysis. We're not going to become all biochemists overnight, but I want to show you what is happening, and I want to tell you the story by using the picture. Okay, so I want you to look at the picture on the right hand side. Let's go over some of this today so we make some progress. The first one is we have glucose, and all you need to know is glucose, the sugar that you eat, candy, the carbs, right, the rice, the bread that you eat, eventually becomes glucose, and it is a 6-carbon molecule. These little black circles that you see over here are representing as carbons, so we have 6 of them, right? So all you need to do right now is look at how many circles there are, okay? It's pretty easy. You don't have to be a biologist or a mathematician to do that, right? So there's 6 circles. The first step is you actually, over here, I want to ask you, do you make ATP or do you use ATP? So I want to ask you this question right here. Go from step 1 to reactant 1 to product 1. Do you use and consume ATP or do you make ATP? All right, that's just a question I want you to think about. We might not answer it today, but I want you to think about that. Now, the P gets added on to the carbon. You see that? The P gets added on. Now, going from the second product to the third product, you split that 6 carbon into 2 3 carbons, right? You guys see that? It's 1, 2, 3 instead of the 6 now. It went from 3, so it went from 6 to 6 with 2 P's to now a 3 with 1 P, and now you have 2 parts. You go through a ton of reactions down this entire chain of 9 to 10 step reaction, and you do a few things. You eventually end up also with a 3-carbon molecule called pyruvate, and you also make some ATP. So what I want you to do now is looking at this picture, and I know it looks kind of confusing, and you're like, oh my god, there's a lot of P's and ATPs and arrows and scary. So what I want you to do is look at the ATP and count for me, okay? So over here we have 1, 2, we have 3, 4, 5, and 6. All right, 6 total. Over here, we actually used 2 ATP. Over here and over here, we actually made ATP. So now the question for everybody is, how many ATP did we gain? What's the net ATP? If we use 2 and we made 4, right? 4 minus 2 is 2. So the answer for glycolysis is, for every 1 glucose, you only make 2 ATP. All right, so that's a number or a ratio for you to think about. So when you're thinking about how much energy can you make in glycolysis, that's roughly what you get. Now, we can take that reaction one step further, and we're going to look at the prep cycle. And this is that brief moment in time when you're actually giving off CO2. Nothing really important is happening except for the fact that you rearrange that 3-carbon molecule, and because you give off CO2, right? Because, so think about this, right? So we have this 3-carbon. So here I'm gonna use that. We'll go back here one really quickly. So if we have a 3-carbon molecule right here, so 3 black circles, and if one of these carbons is given off as CO2, right? This, because there's a C right there, and one of these black dots is a C, we then create something that is 2-carbon, right? Does it make sense? So we lose a CO2, meaning we cut off one of these little black circles. So now if you look at the next page, what's going to make sense is, now what you're doing is you're creating a 2-carbon 2-pyruvic acid molecule by giving off CO2, and then this CO2 loss creates something called acetyl-CoA. This is probably going to be the most significant intermediate in this entire reaction, acetyl-CoA. All right, sounds really crazy, but acetyl-CoA is the intermediate where you can allow different kinds of macros. So if you eat fats and lipids, and if you are on a keto diet, or if you're eating protein, they have the ability to become acetyl-CoA and then go into this reaction. In other words, if you don't eat any carbs, and you eat only fats in your diet, and you eat a lot of protein in your diet, those macros will enter this reaction at this point. All right? And then those high electron carriers, which I was talking about in the previous slide, are things that are like ATP, but they're not fully expressed at ATP form yet. So these 2 of them that are found in our bodies, and they're found widely in all of life, is the NADH, FADH2, NAD positive, FAD. Same thing. All you gotta know is that NADH, right, the H represents a proton, so it can become NAD plus a proton, right? Same thing, right? So it's just a different way to write it, express it. And whenever you see NADH, oops, sorry about that. If you see NADH, or if you see FADH, it's essentially a sibling. It's a cousin that allows you to transport a lot of electrons. And electrons just means energy. All right, so just remember, these are not directly used as energy, they're indirect carriers of energy. Think of them like an ATM. You go to an ATM machine when you need cash, and then you can either deposit the cash when you want to put something in, or you can also withdraw. Okay, so those are your 2 energy carriers. Now, at the very end of the story, you come along to what we call the electron transport chain, and this is where all of the work is done. So remember, in the glycolysis, we only made 2 ATP, right? That's really nothing. We only made 2 ATP in glycolysis, so frown. But in electron transfer chain, we make upwards of about 30. So how do we do this? Well, all you need to remember for the electron transfer chain is that it has about 4 different kinds of proteins and pumps on the membrane. 1, 2, 3, and then there's some smaller ones that you can't see. But what this does is that it moves the protons that we talked about, right, on the NADH and FADH2 to the outside of the membrane, and then it allows the very last pump right here on the bottom, right? So you have one pump number 1, protein number 1, protein number 2, protein number 3, and number 4 on the bottom, which actually will take the protons and drive this pump to join and make ATP. So it's an assembly line that passes the energy down the protein to power this final pump at the very bottom to make ATP. All right, and we are at the 8:50 mark here, so I'm gonna start wrapping this up. But I do want to put a question to the class for today, so you get points. All right, so if you could please stay on just for another minute or so. I'm going to post a question for today, and actually it's the next slide, so perfect timing. So before you leave, please answer this question. All right, so right here, typo right there on C. Which of the following pathways generates the majority of ATP in energy? Oh, ATP from energy nutrients. Oh my God, there's so many typos here. Sorry. So I'll fix the question here. This is bothering me. Typo here. So there was no word check, spell check for this. So go ahead and pick an answer. So that way at least you get points for today. All right.